Slide 1. Factors affecting enzyme activity. Good morning, everyone. Today's lecture, Lecture 5, focuses on the factors that influence enzyme activity. We'll cover key aspects like temperature, pH, substrate concentration, and inhibitors. By the end of this session, you should be able to analyze how these factors impact enzyme function in both industrial and biological systems. Here's an overview of the factors we'll be discussing today. We'll delve into enzyme and substrate concentrations, temperature, pH, the role of activators like cofactors and coenzymes, inhibitors, reaction time, the effects of light and radiation, accumulation of end products, and the presence of water. Each of these plays a crucial role in determining how enzymes function. The rate of an enzyme-catalyzed reaction is directly proportional to the enzyme concentration, assuming there's enough substrate available. This is known as a zero-order reaction, where the rate is independent of substrate concentration. This principle is used in diagnostics, such as measuring ALT, AST, and CPK for liver and heart diseases, and for diagnosing metabolic defects at birth. Increasing the substrate concentration boosts enzyme activity until all active sites are saturated. Beyond this point, the reaction rate plateaus, and adding more substrate won't increase the reaction rate. This saturation effect is a fundamental concept in enzyme kinetics. The study of enzyme kinetics began in 1902 with Adrian Brown's work on sucrose hydrolysis by yeast invertase. Brown observed that at high sucrose concentrations, the reaction rate became independent of sucrose concentration. He proposed a two-step reaction, where the substrate forms a complex with the enzyme, which then decomposes to form products and regenerate the enzyme. In 1913, Leonore Michaelis and Maud Menton formalized these observations into the famous Michaelis-Menten equation. This equation is based on several key assumptions, including a single substrate and product, high substrate concentration relative to enzyme, and measurement of initial reaction rates. It also assumes that the total enzyme concentration is constant and that the enzyme follows saturation kinetics. Let's walk through the derivation of the Michaelis-Menten equation. We start with a typical enzyme-catalyzed reaction and consider the initial state where product concentration is negligible. The rate of reaction is directly related to the concentration and breakdown rate of the enzyme-substrate complex. Remember that the total enzyme concentration is the sum of free enzyme and the enzyme-substrate complex, and substrate concentration is much greater than enzyme concentration. Building on the previous slide, we consider the rates of formation and breakdown of the enzyme-substrate complex. The rate of formation depends on the concentrations of free enzyme and substrate, while the rate of breakdown depends on the enzyme-substrate complex. These rates are governed by specific rate constants K1, K1, and K2. According to the steady-state assumption, the rate of formation of the enzyme-substrate complex equals its rate of breakdown. By setting these rates equal and rearranging the equation, we can solve for the concentration of the enzyme-substrate complex. This leads us to the Michaelis-Menten equation, which relates the initial reaction rate to the maximum rate, substrate concentration, and the Michaelis constant. The Michaelis constant, Km, is defined as the substrate concentration at which the reaction rate is half of the maximum possible rate. Km is constant for each enzyme under specific conditions and represents the enzyme's affinity for its substrate. A lower Km indicates a higher affinity. Vmax, on the other hand, is the maximum rate of an enzyme-catalyzed reaction. The Michaelis-Menten equation provides a quantitative description of the relationship between the rate of an enzyme-catalyzed reaction, substrate concentration, and the constants Km and Vmax. By understanding this equation, we can predict how changes in substrate concentration will affect the reaction rate. Let's consider the case where the substrate concentration is much less than Km. In this scenario, the Michaelis-Menten equation simplifies, and the reaction rate becomes directly proportional to the substrate concentration. This indicates first-order kinetics, where increasing substrate concentration linearly increases the reaction rate. When the substrate concentration is much less than Km, the reaction rate is far from reaching Vmax. Vmax represents the hypothetical maximum rate achievable at saturating substrate concentrations. In this case, the reaction operates well below its potential maximum. Now let's examine the case where the substrate concentration equals Km. Substituting Km for substrate concentration in the Michaelis-Menten equation, we find that the reaction rate is equal to half of Vmax. This provides a direct link between Km and the reaction rate. Finally, consider the case where the substrate concentration is much higher than Km. In this situation, the Michaelis-Menten equation simplifies, and the reaction velocity approaches Vmax. This means that at very high substrate concentrations, the enzyme is working at its maximum capacity. The Michaelis-Menten equation has limitations. The asymptotic nature of Vmax makes it difficult to determine precisely, as achieving full enzyme saturation is rare. Experimental limitations such as substrate solubility and enzyme instability can also affect the accuracy of Vmax determination. Further limitations include substrate inhibition and deviations from ideal conditions. Many enzymes exhibit behaviors that complicate the model. The equation also assumes a simple enzyme-substrate interaction, neglecting multi-substrate reactions and complex enzyme mechanisms. The Michaelis-Menten equation also neglects product inhibition, where the reaction product can inhibit the enzyme. Additionally, it assumes constant Km and Vmax, which can be influenced by changes in experimental conditions like pH and temperature. Slide 19. Each enzyme has an optimum temperature at which it's most active. Temperature increases the kinetic energy of the system, increasing the chances of substrate-enzyme interaction. The Q10, or temperature coefficient, describes how much the reaction velocity changes with a 10 degrees Celsius temperature rise. However, high temperatures can also cause enzyme denaturation. Most human enzymes have an optimum temperature range of 35 to 40 degrees Celsius. The optimum temperature is where enzyme activity is maximized. At high temperatures, enzyme activity is compromised due to conformational changes in the active site. This graph illustrates the effect of temperature on enzyme activity. Curve AB represents the effect of temperature on enzyme action alone, while curve CD shows the temperature effect on enzyme destruction. The observed relationship between temperature and enzyme action is represented by curve AE. Determining the exact optimum temperature can be challenging, as it depends on the measurement duration. However, approximate values often correlate with the body temperatures of the organisms from which the enzyme came. The optimum temperature results from a balance between increased initial rate and decreased active life of the enzyme. The hydrogen ion concentration, pH, significantly controls enzyme activity. It affects the degree of dissociation, electric charge of the enzyme, and formation of the enzyme-substrate complex. Each enzyme functions best at a specific pH value, and activity decreases on either side of this optimum. This slide shows the optimum pH for the activity of various enzymes. Notice how different enzymes have different pH optima, reflecting their specific biological roles and environments. Maximum enzyme activity is often obtained at or near the isoelectric point, pi, of the enzyme. For example, trypsin, with a pi of 10.1, shows maximum activity at a pH range between 7 and 9. The catalytic activity of enzymes can be altered by modifiers, which can be inorganic or organic molecules. Positive modifiers, or activators, increase enzyme activity, while negative modifiers, or inhibitors, decrease it. Some enzymes require a metal ion for full activity, and removing the metal can lead to loss of activity. Metal ions can act as activators in several ways. They can directly participate in catalysis, especially in oxidation-reduction reactions, by undergoing valence changes. For example, iron functions in cytochromes or catalysts. Sometimes, a metal combines with the substrate to form a metallosubstrate, which is the true substrate for the enzyme. This forms an enzyme metal-substrate complex, which then decomposes to produce the reaction product and regenerate the enzyme and metal. Metal ions may not directly interact with the enzyme, but instead interact with the substrate, making it able to interact with the enzyme's active site. For example, magnesium ions are required by all ATP-using enzymes, such as hexokinase. A metal ion may first combine with an enzyme to form a metalloenzyme, which then combines with the substrate, forming an enzyme metal substrate complex. This is another way metal ions can activate enzymes. Metal ions can also induce conformational changes in the enzyme molecule, converting it into an active form. In this case, the metal may be linked far from the substrate and maintain an active tertiary or quaternary structure. For example, manganese binding to HIV-1 integrase induces a conformational change, increasing its activity. Recent advances in enzyme inhibition include slow-tight inhibition, where the enzyme inhibitor complex undergoes a conformational change to bind more tightly, and substrate inhibition, where activity decreases at high substrate concentrations after Vmax is achieved. Slide 34. In product inhibition, the end product of a multi-step reaction inhibits the first or regulatory reaction of the pathway. This is a form of feedback regulation where the end product controls its own production.